Our next speaker is Nipam Patel, Director of the Marine Biological Laboratory and Professor at the University of Chicago. He also received his undergraduate degree from Princeton, and he received his PhD from Stanford. Dr. Patel is the director of a storied laboratory that studies sea creatures, but his talk today is about butterflies and the science behind their beautiful patterns and colors. An expert in evolutionary and developmental biology, he focuses on how animals develop the body shapes, patterns, and coloring that make them distinctive. His lab's website sums up the purpose of his group's research in one sentence. Our goal is to find the connections between color, genetics, and the way life emerges as a result of the two. Elected to Class 2's section on cellular and developmental biology, the title of his talk is The Physics and Development of Beauty, Structural Colors in Butterflies.
Thank you all for this opportunity to speak here today. So most of my lab is really interested in the evolution of development. So I'm trained as a developmental biologist, and one of the things I'm interested in is how animals have evolved into different morphologies and different patterns of development. And here you see some of that variation in a very animal-centric slide. But I'm going to tell you today about some newer work that's gone on in the lab. But I want to start this story way back in time. So this is a tiger swallowtail butterfly. I was 8 years old, and I found this butterfly dead in my yard. And I took it in, and I had a book called How and Why of Butterflies and Moths. And in the end of it was some explanation about how to mount butterflies. And so I mounted this butterfly very poorly. This butterfly is still in my collection. It's actually still at my parents' house. But very shortly after I put this butterfly and mounted it, I convinced my mom to start sewing butterfly nets for me, and then my dad let me take a hacksaw and hacksaw some of his golf clubs to make the shafts for the butterfly net, and I started collecting butterflies, and it's a hobby that I've continued ever since. So fast forward 55 years, and this is part of my collection. This is, I'm embarrassed to say, 0.1% of my collection. So I have a very large butterfly collection, and I sort of jokingly say that, well, at a certain point, I had to justify to my wife why I have this giant butterfly collection. And so I decided it'd be great to really, bring my knowledge of developmental biology to bear on some interesting questions in butterflies. So there's great research going on in the genetics of patterning these butterflies, but I decided to pursue a slightly different question. And that's how they get the color that you see that they have. And in particular, I'll tell you about how they make green and blue, because it's a little bit different from the way that you might think about it. So where does the color come from, right? So if you zoom in on the wing of a butterfly, the butterfly is covered in scales. So these scales look very small to you. A typical scale is about 300 microns long, maybe about 50 microns wide. But they're an extension of a cell. So they're actually a giant cell. This cell grew while the butterfly was a chrysalis. And then it got covered in chitin, which is the sugar polymer that covers the surface of all arthropods. So that's a very tough, hard material. And then the cell died, before the butterfly or as the butterfly emerged. But the colors come from two different mechanisms. One is pigments, right? So the long wavelengths of the rainbow, so colors like red and orange and yellow and things like that, are generally made by pigments, right? And what are those? So they're individual molecules that absorb a particular wavelength of light, and then what's reflected gives you the perception of color. So for example, that yellow highlighter has that organic compound in it. That's particularly good at absorbing blue light. If you absorb blue light out of a spectrum of white light, what's reflected you perceive as a yellow color. So that's how pigments work. They're individual molecules that absorb a particular wavelength of light and give you color. And that's the way that many colors are made in living organisms. But it turns out for short wavelengths of light, so short wavelengths of color, so green and blue, right? What you have to do is absorb red light. And it turns out that most biological systems have a difficult time making molecules that are good at absorbing red light. There's a big exception in the world, which is chlorophyll, which is why the world is green, but it's a somewhat unusual system. So how do you end up doing this, right? And it's not just a problem for butterflies. This occurs in sort of all living biological systems. that in fact making green and blue is an interesting problem that usually in fact is not solved by a pigment. Instead, it's solved by a phenomenon of light refraction. So the easiest way to think about this is to think about something like a soap bubble, right? So the soap doesn't have any color, but the soap bubble has all the colors of the rainbow. So how does that happen? And it happens because the thin layer of soap, right, that material has a high refractive index, so light reflects off both the top and bottom surface of this thin soap layer. And then remember, light has these wave-like properties. So what happens to those light waves is then they can either interact in various ways. So if they're in phase, they can interact in a constructive interference and double in amplitude, look 4 times brighter to you. If they're out of phase, then that wavelength gets canceled out. Furthermore, remember that white light is really a spectrum of colors, and then each of those colors has a different wavelength. So what that's going to mean is that when light interacts with a thin material like this, you can calculate those wavelengths that undergo maximum constructive interference with this simple equation, and the wavelengths that undergo maximum destructive interference by removing that 1/2 term in the equation. And so what's going to happen is that you're actually going to get sort of curves like this. So for a given thickness of layer, called the lamina there, you'll get a particular spectrum of wavelengths that are either constructively amplified or destructively decreased, and so you'll have a characteristic color. So at around 200 nanometers thickness, you'll have that spectral curve, and you will observe that as a blue color. So you can create color that way. So you can amplify that effect if your reflective layers are stacked one over another. Okay. So what I'm going to show you in the next picture is a picture of a moth, and in this moth, the only pigment color is black. Okay. But it can make all the colors of the rainbow, okay? And it does that because the scales are multi-layers of chitin and air. And they're tuned to different thicknesses, so they can be all of the different colors that you see on the rainbow. And so that's one way that, in fact, you can make all the colors by a structural solution or this refractive light solution. Here's another great example. So these blue morpho butterflies that come from Central and South America, they have this fantastic metallic, very, very bright blue color. So how do they do this? If you look at any scale, this is just a typical pigmented scale, they have this great architecture to them. They're all, again, all of this is made out of chitin. And in yellow are the ridges that scales have. And what happens in morphos, if you look at their scales, at first glance, they look like every other scale. They've got ridges along their length. But if you do a cross-section across those ridges, you'll see that they're stacked up into this Christmas tree-like structure. And the thickness of those branches and the spacing of the branches is just right to get this massive constructive interference of blue light, so they appear this incredibly bright blue color. Another example is shown here in these little hair streak butterflies that are green. Now, green's a really useful camouflage color for an insect. But only a few genera of butterflies has ever invented a green pigment. Otherwise, all the solutions are structural. So what happens in this particular species? So if you shine white light down onto the scale, what you see is this green light coming back at you. But you notice it looks sort of like crystals inside of the scale. If you break one of these scales open, what you see is this honeycomb-looking material inside of the scale. So it's not the ridges that are doing this, but this honeycomb. But this honeycomb has a very specific shape called a gyroid. So in 1970, there was a mathematician, Alan Schoen, who came up with a set of mathematical equations that defined a triply periodic minimal surface that divides space into 2 non-overlapping domains. And lo and behold, people discovered that it exists inside of the scales of certain species of butterflies in order to make them a green color. And butterflies have evolved this 3 times independently as a way to be green. Okay, so those are all discoveries made by optical physicists who, do the same thing. They get a dead butterfly out of a collection, they section it, they work out what the shape and the geometry is, they do the math, which I can't follow half the time, and they say, well, this is why it's this color. Occasionally they have a lot of money, they'll make it out of man-made material, but in the end, their papers always sort of end with a paragraph that said, wouldn't it be great if someone figured out how the butterfly does this? Okay, so that's the quest that we're on in the lab, is to really try to understand, well, how does a living system make something like this? How does it make nanostructures? And how can it make it so precisely? Because if these things are 10 nanometers the wrong thickness, they're the wrong color. They don't work for it, right? So that's what we wanted to do. Now, one of the challenges is that all of this is going on while a butterfly is a chrysalis. And if you've ever looked at a butterfly chrysalis, can't really see into it, right? So one of the first things we did is developmental biologists we really are very interested in watching these things live, watching how they're making these structures. So we developed ways to be able to make windows into the butterfly chrysalis. So here you're seeing a butterfly that's just pupated. And now you can see 8 days of development, and you're going to see all of wing development. So you see the wing veins are already there. And now when it turns a sort of silvery color is when the scales are first growing. You'll actually see a little bit of structural blue color in one edge of the wing. But now towards the end of this period, you will see all the pigment colors come in. You'll have a perfectly formed wing, but now we've been able to watch all of wing development. Okay? So now we can go in and actually see the scales growing in great detail. So what you're seeing here are scales, and the fluorescent signal is filamentous actin. So we've shown that this filamentous actin is a cytoskeletal network inside the scale that's very important in setting up the pattern of the ridges of the scale. So now we can see how they're formed in normal scales that are just pigmented, and ask how that's modified in order to make some of the nanostructures or larger scale structures that are required. Here's a more closed-in, close-up view. So you can see now that we can actually watch these scales growing during these important stages. And our goal now is to really understand how the nanostructures are being produced as these scales grow. The other approach we're taking is a genetic approach to try to understand how these colors form. So these are buckeye butterflies, a very common butterfly in North America. And you would say, well, this is a terrible choice for studying structural color because it's all the colors I told you that were pigment colors. It's brown and orange and white and so on. But if you collect buckeye butterflies, you notice that a small fraction of them in North America have a little bit of blue on them. So you can see in the leading edge of the forewing, that little blue area. And there's a woman named Edith Smith in Florida, and she ran a butterfly company that would raise chrysalises to then sell to the big butterfly houses that you can walk in and see live butterflies flying around. She also noticed that buckeyes have a little bit of blue, and it occurred to her, what if I breed the bluest ones together? And so she did that as sort of a hobby for a year, which is about 12 generations, and she made them shockingly blue. Okay? And we were buying butterflies from her, and we got some of these, and they came out, and we were like, what are these things? And so we called her, and she said, oh, do you like them? I made them. And so she had done a huge amount of work for us. She'd done a selection experiment, a breeding experiment. And Rachel Thayer was the graduate student in the lab that started studying these. The first thing that Rachel showed is that the reason that they're blue is the bottom layer of chitin, the lamina, is what's blue. So if you look at the scale from above, it's not the ridges that are blue. If you flip it over, you see them really well. And if you scrape the ridges off from the top side, you see the blue color really well. So what had happened is that bottom chitin layer of the scale had become just the right thickness to become a constructive reflector of blue light. But where did that come from? So if you look at the original butterflies, the brown butterflies, they're actually gold. So they have brown pigment in them, but they also have a lamina that's very thin that's giving this long wavelength reflections of this kind of yellow light. So what had happened in the selection is the lamina had gone from about 100 nanometers to about double that thickness. And so that's what had happened. And there are actually naturally occurring, closely related species to the buckeyes that have natural blue and green colors to them, and this is exactly what they've done. So the selection experiment recapitulated also what had gone on in evolution. So now the beauty is that now we can use genetic mapping to find the loci that were selected in this experiment in order to make them blue. So the idea is we cross the brown and the blue butterflies together. So the F1 generation, the children, right? So for each of those butterflies, one chromosome copy comes from the brown parent, one from the blue parent. The F1s look pretty uniform. They're in between the two parents, right? But in the next generation, if we breed brother and sister together, right, and we get the grandchildren now, because of chromosome recombination that occurs now, these butterflies contain regions of chromosomes that come either from the brown grandparent or the blue grandparent. So we can sequence these F2 individuals, right? And we can phenotype them. And then we can look for those genetic loci in the genome that are actually contributing to that variation and giving this blue color. And Rachel was able to raise fairly large families in the F2 generation. I didn't actually know that a female butterfly could lay that many eggs, much less that someone could raise them all to adulthood, but she did. And so the variation in F2 is really beautiful. So you have different amounts of the wing with iridescence, and you have lots of different hues, right? Ranging from blue through, turquoise and green. And so if we just map the percent of the wing that has this structural color to it, what we see, so the linkage groups of the different chromosomes, and those peaks show that there's a statistical significance of a locus in that area that has a strong effect on the color. And so you see there are two big peaks here. And in fact, we know what one of these genes is already. It's a gene called optics, it's a transcription factor. And in fact, if we use CRISPR to genetically engineer the gene, so it was first described because it controls the red color in heliconia, shown on the right-hand side there. But if we use CRISPR to knock it out in buckeye butterflies, in fact, they turn blue. So that is definitely one of the loci. We don't know what the other peak is. But maybe more interesting is to actually, instead of using the percent coverage of the wing by structural color, use the hue as the phenotype. So this is going sort of from indigo to blue to turquoise to green, and this is a difference in thickness of the lamina from about 190 nanometers to 240 nanometers. So what are the loci that can precisely control that thickness? So if we look at that, what we see is, again, there's a couple of different peaks. There are loci that have a very strong effect on this. We don't know what any of these are, but that's what we're really interested in, is using genetics to answer the question of what are these genes that can so precisely control the thickness of this particular layer, okay? So with that, I'd like to thank the people that have done all this work and guided my career. So this is my lab through the years, and they're just an incredible group. And then these are some of my mentors. So Mal Steinberg, who was my mentor at Princeton, Corey Goodman, who was my graduate student advisor. But I also want to highlight these other two people who are my high school teachers, Mrs. Foster and Mr. Ray. And so I actually took embryology as a high school student. I also learned microbiology from Berge's determinative bacteriology, and biochemistry out of Scientific American articles. And it was a public high school, and I was exceptionally lucky. And lastly, my family. So my parents that encouraged me in everything that I do, my extended family, my wife, and all of the colleagues that I've had through the years teaching at the MBL. And thank you very much for your time.
Beautiful talk. Do we have any questions? Rich.
Hey, Nipam, Rich Mooney at Duke University. So in the example of the morpho, where there's structural pigment on the top surface and cryptic coloration underneath, I'm just wondering about predator versus mating selection for these different classes of pigments and whether there is a rhyme or reason to that.
Yeah, I don't know that there's a rhyme or reason. I mean that, as you might expect, that natural selection in the environment really is the selective pressure for that. But the morphos are a great example. So on these morpho butterflies, right, the bright blue is on the upper side, right, and they're cryptically colored on the underside. And if you've ever chased them, you know what they do. They flap that blue color, you start visually focusing on that. And then if they're being chased, they suddenly sit down and they won't open their wings, they'll hold them closed. And so your mind keeps looking for this flashing blue and you walk right by them, right? They're sitting there. So they use that, but of course the males are using it as a show of display to each other for mating competition.
David Weinberg, Ohio State University. So artists are always trying to find unique colors like Yves Klein blue or something. So are there examples in art that you know of where people have actually used these interference or scale things rather than pigments for getting colors?
Yeah, you absolutely can. So like you can make paints out of colloids instead, and they can actually have color not from pigment but from this sort of iridescence effect. And you can imagine there's a lot of interest. For example, if you could make paint that was functioning structurally and not by pigment, then it wouldn't fade in the light. And so it would be much more resistant. So there are a lot of applications, but for us, it's just really trying to understand what the butterfly does here.
Hi, I'm Bill Bialik from Princeton, Nipam. Great talk. When you see a bunch of structures next to each other and they're all the same, do you know whether each one is being instructed to get the right scale, or whether they're interacting with each other and kind of crystallizing into that pattern?
Well, I mean, those are questions we would love to be able to answer, right? So some people ask, well, are these emergent properties of the structure? So do you have a biophysical, mechanism that just gets them to fold in the right way, and you don't really have to specify them? And that would work across, ridge after ridge. Or how precisely do you have to pattern them? And we don't know the answer to that, right? So one of the intriguing ones is the gyroids. So the way they actually form is that the ER actually first folds, the endoplasmic reticulum first folds into a double gyroid, and then you basically secrete chitin into that. And you can actually, by misexpressing proteins, in the ER, you can get endoplasmic reticula to fold up into all sorts of structures that are like gyroids. So that may be that it's just sort of an emergent property when you have the conditions just right to make these things.
Thanks.
Terrific, thank you.